and Mondays. I think I just kind of forgot. So questions you have for the pending doom today, right? Chapter 6 and 7, and on to chapter 8, if any of you are ready for that, which I'm doubting, but we'll just go with, how about questions from 6 and 7? Come on, Stephanie, I know you have a question.
I know, give me a minute, I'm sorting through my notes.
All right. Anyone else while we're waiting? No, yes. What about the cytoskeleton, Bo? What do you want to know about it? Who else has joined? Is it only the animal cells? No, actually it's not. You'll find the cytoskeleton in, what you will only find in mostly animal cells would be, I meant to do a blank screen and to draw. So the cytoskeleton, Stephanie and I had this conversation a little bit yesterday during one of my office hours, right? So this chapter, all of chapter 6 and chapter 7 for that matter, they are, we're talking about generalized cells, and these cells don't really exist in the real world. It's only this manner that we teach you all the possible structures that might be in a cell, right? So those generalized pictures of an animal cell and a plant cell on pages 100 and 101 are just that. They are pretend. That's why they're drawn in cartoon. That's why they show you other cells along the bottom and highlight some of the key features of actual real cells, both with a transmission electron microscope and a scanning electron microscope, to kind of show you what you can see inside a cell with just a regular light microscope. And again, that's why they make them look cartoony in the pictures, because there isn't really a cell that looks like this on these pages. What is it, 100 and 101? They don't really exist in nature. So if we go back to the cytoskeleton, which was your exact question, I get it. So those cells, they show up in eukaryotic cells, and this is a good time for us to talk about this. I'm going to put mostly in eukaryotes, because it's time for you guys to stop thinking about plant cells and animal cells as being the different kinds of cells, because they're not. Plant and animal cells are almost the same, minus a few structures and obviously their functions. Mostly in eukaryotes, remember, eukaryotes, that whole domain, eukarya, right? That includes protists, which are all the single-celled organisms like Euglena, algae, Chlamydomonas, which was in there as an item, as a picture, Trichomonas, Giardia. I'm trying to think of all the protists that you might have some recollection of. A Paramecium, an amoeba. I know I've mentioned those two already. Euglenas. Those are things that you might have encountered. All the algae in the ocean pretty much are protists. All the plants, all the fungus, all the animal cells, right? And you are a eukaryote. That will help you remember that, right? You are a eukaryote. All the animals, all the plants, all the fungus, all the protists. The other major type of cells is not plants. The other major type of cells are prokaryotes, which is a big topic in the beginning of chapter 6, and probably in the video I kind of made a big deal out of it. And believe it or not, prokaryotes, some actually have some parts of cytoskeleton. We used to say they didn't, now we say they do, because they have microfilaments, so actin filaments that are similar to cytoskeleton that appears in eukaryotic cells, in some protists, in some prokaryotes rather. So no, not just in animal cells, right? All eukaryotes have some elements of the cytoskeleton. Does every single plant cell have all of the components of cytoskeleton? No, right? Cells that have flagella, which are going to be many of the protists, some of fungus, and a few actually simple plant cells have flagella, right? All of those cells that have flagella or cilia, they're going to have obviously microtubules at minimum, right, to form those flagella, as well as cytoskeletal components. Some protists don't have a cytoskeleton on the inside. They have some of similar elements wrapping them around them on the outside, like we call it a pellicle. What is important for you at this juncture for Bio 1 to know about the cytoskeleton is pretty much what occurs at table 6.1. You need to know those different types, different components of the cytoskeleton, and basically what their, you need to be able to describe them, and then what their function is in a cell, in a eukaryotic cell. So the microtubules have the most varied function, right? They're used by cells for all kinds of things. And I would just hit the highlights that I mentioned in the video about chapter 6, as well as what's on the outline from chapter 6, as far as the components of the cytoskeleton and their main features. And again, you're probably going to find that mostly in the video from chapter 6, because I go through and say, you don't have to know all of this, but you must know at least these 3 things or whatever it is. So that table on page 113 in your book, this table 6.1, that's probably the best condensed version of what you need to know about the cytoskeleton. They do go on and on about the microtubules, particularly in flagella, and how the flagella work with the dynein arms and the ATP, and how they grab the central, how those radial components grab that central part and then cause the flagella to twist, which causes it to move in a wave. All of that, I don't care, not important, right? That's kind of blah, 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 blah. That's a little too much detail. I'm not going to ask you that. But you do need to know what the components of a flagella and a cilia are. You do need to know that they're made of microtubules and the characteristics of microtubules. Absolutely. Perhaps what you were thinking, Bo, and I'm not trying to put words in your mouth, but perhaps what you were thinking is the extracellular matrix, or extracellular, your book calls it, yeah, the extracellular matrix. That's an animal thing, okay? That's predominantly, and I would say in multicellular animals, because there are animals that are single cells. And I'm going to put mostly, because it's never only, right? We are all cells are derived from a set of common ancestor cells that contains some various features. And so it's never an only. There is, except like if you're talking about sharks, right? And they're the, chondrichthyes, and you're only talking about their particular unique characteristics. In Bio 1, since we're talking about cells, right? And we're talking about eukaryotic cells predominantly, a little bit about prokaryotic cells, but mostly eukaryotic cells, it's hardly ever going to be an only, right? So that extracellular matrix and the extracellular fluids, that comes up with the animal cells, talking about the end of chapter 6, right? And those cell-to-cell junctions, those also mostly animal cells. I'm going to say animal cells predominantly. And remember, in plant cells, we have the plasmodesmata, we have the middle lamella, we have other ways of extracellular matrix that the plant cells can communicate with each other. Okay? But the extracellular matrix in particular, we would say, is pretty unique to animal cells. And those cell-to-cell junctions, the tight junctions, the desmosomes, and the gap junctions, those are discussed just in terms of animal cells, right? And the examples given are in terms of animal cells. And that, again, preview you a little bit and get you a little bit ready for anatomy and physiology, because those cell-to-cell junctions are going to be different in different types of integument, different types of connective tissue, different types of muscular tissue. And those words are going to come up when they're talking about cardiac muscle, for example, and how it's different from skeletal muscle. One of the differences is going to be the type and amount of different of those cell-to-cell junctions, how many gap junctions versus how many desmosomes. So these are things that we're trying to introduce to you so you have just some vague recollection when you get to anatomy and physiology that you've at least heard these words before. You'll forget, and if someone asks you point blank, you'll say, no, I've never heard of that before. But rest assured, we know that you covered chapter 6 in Bio 1, so you've at least heard those words and memorized some definition about them sometime in your past, right? So that's more than you were after. Let's just minimize this. Can I do that? No, I have to. Let me see if I can just make me big here. I guess I have to stop sharing in order for that to happen. Let's say pause the share. Let's see if that works. You're welcome. Like I said, that's probably longer than you wanted, but generally that's what I can tell you about the cell-to-cell junctions and cytoskeleton. Other questions you guys have from 6 or 7?
I'm ready.
Okay, Steph.
So I understand that the hypotonic solution swells and can burst, the isotonic stays the same, and the hypertonic shrivels. Where I'm getting held up on this is how you know what type of solution it's going into. Do I need to know that at this point?
Yes, the short answer is yes. So just real briefly, right? So those designations, hypertonic, isotonic, hypotonic, refer almost exclusively to just osmosis, okay? So osmosis, remember, is the diffusion of water, and I'm gonna put that in all capitals because it's only the diffusion of water. It's not the diffusion of gas. It's not the diffusion of salt. It's only the diffusion of water. It's only the movement of water across a membrane from a higher concentration of water to a lower concentration of water until equilibrium. Right? So we're humans, we can't help it. We want to generalize, we want everything to be the same. It's not, okay? So this is a form of passive transport. No water, sorry, or can't talk and type, nor can I talk and write on the board at the same time anymore. If I'm saying different words than I'm actually writing, but nonetheless. Type of passive transport, so no ATP required because we are moving down or with the concentration gradient from a higher concentration of whatever it is, in this case water, to a lower concentration of water. So what I tell students is this isn't the only way water can move across the membrane, right? When you get to anatomy and physiology, you're going to learn about other ways and other channels that move the salt from one side to the other, right? We're just talking about osmosis right now. That's only the diffusion of water. So focus on the concentration of the water. That's the important part, the concentration of the water. Where is the water concentration, higher or lower? Oh, it turned all of them red. I thought it was gonna just turn the ones I was highlighting, but alas. So when we draw those beakers, which I'm gonna make them a little skinnier here, and see if I can draw. All right, so when I draw those 3 beakers, which I would much rather draw on my chalkboard or my whiteboard, but I'm too lazy to go upstairs to the Zoom room. So here are the beakers, the containers of water, whatever you want to think of them, right? And because it's water, of course, it is an advantage of doing it online is I can use more colors, right? So here are the solutions. And notice that I've made the solutions all the same volume because it's not about the volume. It's not about the space the water is taking up. It's about the concentration of the solutes in the solution. So where is there a higher concentration of water versus a lower concentration of water? And my advice, and I know this is not probably what you've been taught before, but my advice is, and I think I've said it in the videos for the lecture, there are lots of things, especially in Bio 1, when we're first learning these concepts, there are lots of things that are opposites. And it makes us sound important, right? If we can describe one of them and then describe the opposite one. And that's confusing to students because it's too many words they don't really understand or know, like committed to memory. And so it's not helpful to give them lots of different ways sometimes to think about something simple, right? So what I tell students is memorize one of them. Just memorize one of them. So don't even think about what hypotonic means. Just focus on hypertonic or the opposite, you pick. And then once you know what that one is, just memorize that one. And all you have to remember then is the opposite word means the opposite thing. And your brain can do that work even in 45 seconds when you're taking the quiz, okay? So it's important to not try to memorize all of these opposite definitions and keep them all straight because that's very difficult, especially at the beginning. So I would advise students all the time when you're studying, if you come across things like this, like pinocytosis and phagocytosis, passive transport, active transport, right, in big categories, hypertonic, hypotonic, just pick one of those words, one of them to memorize and really understand. And then you don't have to memorize the other one because your brain can puzzle it out. All you have to remember is, oh, that's the opposite. So if hypotonic is this, then the opposite is that, right? So that's just a little study tip for you for pretty much the rest of the semester, because oftentimes you have to learn these things. Oxidation reduction, just remember one of them. And then know that the acids and bases, just remember what an acid is. And then anything that doesn't match that, it's got to be a base, right? Rather than trying to memorize all the characteristics and all the words, all the vocabulary that go with acid and all the vocabulary that goes with base, right? So that's just help you a little bit with your studying. I realize it's probably too late now because the test is today sometime, but anyway. All right, back to your original question, Stephanie. Sorry, I was sorry for the digression there. So osmosis is a type of passive transport. So we have, let's make this solution, that's kind of a big box. Let's make it a little smaller. Well, all right, so let's make the first one the hypertonic, and then isotonic, and then hypotonic. All right. So now those words can be used, those words can be used to either describe the solution or the cell, inside the cell. So I guess I can't do that. So let's just make this a small box and put hypertonic here. Okay, so this is going to be the hypertonic solution. Okay, so what does that mean, right? So hypertonic solution. So a solution, so we have to go back to chapter 2, right? A solution is going to be some sort of a solute, which is almost always water, or sorry, some sort of a solute, something, in this case, we're going to use salt. Sodium chloride, our friend, right? But we can add, it can be anything. It could be, the solute can be sodium chloride alone. It could be sodium chloride plus glucose. It could be a lipid. It could be, lipids aren't very soluble in water, so that might not be a good example. It could be a protein. It could be a peptide. It could be a set of amino acids. All of those things can be solutes, as well as any salt, not just sodium chloride. And you have to have a solute and a solvent. And in the case of most of our solutions, the solvent is water. Remember, water is the almost universal solvent. That was like the fourth special characteristic of water, right? And that's where this is coming back to haunt you again, right? Already from chapter 2 to chapter 7. So a hypertonic solution then has to do with, and I'm going to draw this as an arrow between these two because this is important, and students often don't make this connection, right? The hypertonic solution has to do with how much solute compared to solvent. And it's always compared to another solution across a membrane. So these types of solutions that we're talking about, hypertonic, isotonic, hypotonic, they are all relative compared to the other side of the membrane. So in my first beaker here, I'm going to draw my cell and I'm going to make it kind of large, right? Phospholipid bilayer on this cell. So phospholipids plus cholesterol, we'll make it a plant cell. An animal cell. Cholesterol, proteins sticking in there, transmembrane proteins, carrier proteins, all kinds of stuff. Inside a cell, relatively, and I'm making this a little bit low on purpose, we would usually say inside the cell, the cytoplasm is a concentration of between 80 and 90+% of water, right? But I'm going to make it 80 on purpose because I'm going to be trying to illustrate. So 80% water inside the cell. That's hard to write. There we go. 80% water inside the cell. So what that means is all the other stuff inside the cell, the nucleus, the endoplasmic reticulum, all the salt, all the glucose, all the polypeptides, all the amino acids, all the lipids, all of those things that are inside the cell decrease the space for the amount of water that can be inside that cell, right? They're all dissolved, if you want to think about it, in the water. Now, a hypertonic solution, there is a higher concentration of water inside the cell than outside the cell. So in my hypertonic solution, I am going to put only 50% water. So if there's only 50% water, and I really can't keep doing that, so I have to make my little box over here. If there's only 50% water in my solution, that means there's a lot of other stuff in that solution, right? So if the solution equals 50% water, then what? Okay, it has to be 50% something else. So let's say there's 10%, let's make it 15%, dissolved glucose, and maybe other monosaccharides. And maybe we have another 15% salt. All right, so we're up to 30% dissolved stuff, right? Maybe another, let's just keep it easy. Let's make it 20% amino acids. And this is not obviously a real solution, right? So I'm making up. So the solution is 50% water and 50% dissolved stuff. Inside the cell, there's 80% water and only 20% dissolved stuff. Osmosis, my box disappeared when I hit the erase button, but if you still have that written down somewhere in your notes while I'm talking, right? Osmosis, remember, is the movement of water across the membrane from a higher concentration of water to a lower concentration of water. Not volume, but concentration. So where is there a higher volume of water? Where's there more percentage of water? Inside the cell, not volume, percent of water. Inside the cell or outside the cell? Concentration of water. You get a 50-50 chance. I vote both ways. How many people for more water inside the cell?
I'm with Sal.
How many people for more water outside the cell? And I vote twice so that you're not swayed just based on my vote, right? So yes, Stephanie, you're correct. There's more water inside the cell. So where is the water going to go? The water is going to move from a higher concentration of water to the smaller concentration of water until we hit equilibrium. I meant that to be blue. So the water is going to move out of the cell, right? So it's two things you have to think about. Water is moving from the cell to the solution. And what the consequence of that is the cell will shrink. Period. In nature, this does not exist unless you are in the desert and not drinking any water. Or let's say you just had a bout of food poisoning and you had vomiting and diarrhea and you're dehydrated. Or you're a plant and no one's watered you and it hasn't rained for a month. This is not real. This is not a usual situation. And cells, because they're living things, right? Back to chapter 1, themes of biology, they have ways to regulate this. So when you learn about the kidney in anatomy and physiology, or you learn about blood and transporting molecules across the membranes in blood, that's what this is all about. It's about how the body, how an organism compensates so that this doesn't happen, right? So we need to teach you about these types of situations, what could happen, before we can teach you how your cells respond to the environment, right? How they regulate those characteristics of living things inside a whole working organism, okay? Giving me a lot of air there, it's a little better. So in a hypertonic solution, the water is going to leave the cell and go into the solution until it hits equilibrium, and the cell will shrink, right? If that gradient is high enough, right? Maybe if the water, if it's only 30% water, for example, if that gradient is high enough, yes, you can eventually kill the cell, right? This is what we do when we make jelly and jam and we use sugar, or when we make, partially when we make pickles, we use acid and salt, right? To increase the concentration so much the water leaves the cell and preserves it, because the water is leaving the cell, then the mold can't grow on it, right? That's a discussion from microbiology. Come to microbiology, learn more about it. Now we're going to look at isotonic next. And I know you don't want to, you want to look at hypotonic, because you're like, I understand isotonic, but we're just going to make sure here, right? So I'm going to put a little box here and put iso, right? So isotonic solution would be, and I'm going to take the painful minutes to draw that, right? Isotonic solution would be when I have 80% water inside my cell and 80% water in my solution. So I've decreased all of these items over here, right? So the solution would have less of all of this stuff in it. Isotonic, remember, means the water is still moving. It's just moving one for one. So when one water molecule moves in the cell, one moves out. The membrane is always busy. There are things always going on in that membrane. It's not that it's never moving. It's that it's moving one in, one out. It's at equilibrium. Now, again, why do you care? Excuse me. Because let's say you go to the hospital and you get a bag of fluids, right? Because you're dehydrated or you're sick. That's what that is. It's an isotonic solution, as close as we can get to what's in your bloodstream, because we don't want to shrink your red blood cells or explode your red blood cells, because then that wouldn't help you, right? So there's an example of the use of an isotonic solution. All right, now last but not least, the hypotonic solution over here. So again, remember, it's comparing the concentration of one side of the membrane, the solution on one side of the membrane versus the other side of the membrane. So I'm going to draw my cell again, and I'm going to leave it at 80% water inside the cell, right? But now outside the cell, I'm going to put 100%. This is going to be pure water. And I'm going to have to probably buy that, or I'm going to have to double distill and deionize it in my laboratory, right? Not from a bottle, right? That's not 100% water. That's just stupid. Stop drinking water out of bottles, right? You all live where the tap water is fine. You can just put it in a container and carry it with you to school. Just being lazy. Plastic bottles, right? We're trying to get rid of those. Bad for the environment. Again, I digress. My apologies. So in a pure 100% solution, a hypotonic solution, now the water, let's see if I can get my text box back here and turn it blue, right? So in a hypotonic solution, there is a higher concentration of water outside the cell on that side of the membrane compared to inside the cell. So the water moves in because it moves. Water's always moving from a higher concentration to a lower concentration. So, oh, I like this brick color. Let's use that. So now the water molecules, more water outside the cell, higher concentration outside the cell than inside the cell. Water is going to go into the cell. What is the consequence of that going to be to my cell? Right? The cell will swell. That's easy to remember. Look at me writing with my right hand. The cell will swell, right? Opposite of the cell will shrink. So again, just remember one of them and then puzzle out the other one if I ask this question instead of that question. Again, not a situation in nature. This doesn't occur. Don't believe what you've read on the internet about people drinking too much water and dying from being in a hypotonic condition, right? That's almost impossible to do, right? So don't actually believe that. Again, there are mechanisms in your body to control for this, right? This is, you take a cell and you put it into, and again, why do we care? Because what we're going to find out is a lot of scientists, researchers, medical people, they study cells outside of their natural environment. So it's really, really important that we understand this because we want to study the cells in this situation when they're isotonic, right? We want to make them as happy as possible so that they'll do their activities and reveal their interesting skills to us as a researcher, right? These situations, again, are super extreme and don't often happen in nature unless there are extreme situations, right? And again, in anatomy and physiology, they're going to be talking to you a lot about how the body responds to being slightly hypotonic, what happens in a particular organ or organ system, or slightly hypertonic, or why that is useful in a particular situation in the body, like when you're trying to excrete waste. So that's why you have to learn this, because it's going to keep coming back again and again in your more advanced classes, right? Try as hard as you can to remember that these words, these ick words, it's relative. It's relative. You're comparing this solution to the solution across the membrane. Now, I'm going to say something that it's not important to memorize for the test, but I just want you to think about a little bit, try to work those cerebral brains of yours, right? In this diagram, the cell is hypertonic to the solution. The solution is hypotonic compared to the cell. That's what I mean by relative, right? It's relative concentrations across the membrane. And remember, the cell will swell unless the concentration gradient is so great that water keeps coming in and it's going to burst. Again, most cells have mechanisms to prevent this from happening, right, to the bursting level. And plant cells, like they show in your textbook, plant cells prefer to live in a hypotonic environment because that helps keep their turgid state, right, their turgidity, their rigidity. It helps the plant stand upright because it's all the cells are filled with water, right? So again, it's relative and it depends on particular situations, and they probably show you a picture of a paramecium or something. Yeah, there's a paramecium squirting water out because the water keeps coming in and the paramecium can't do anything about it. So it has a little pump that just squirts out the excess water so that when it gets too hypotonic, the cell doesn't explode. It gets rid of a little extra water, the water keeps coming in and it squirts back out, right? They're showing you that as an example of how cells have managed to, I want to say, I guess, deal with or evolve so that they can live in a hypotonic environment, right? Anything that lives in fresh water is living in a hypotonic environment. Again, probably more than you wanted to know. Anything particular before I leave this picture?
Thank you.
You're welcome. Anybody else? Questions, comments, worries, concerns for the pending Doom? Yes, no? All right then. I'll be adding materials and probably adjusting some of the dates for some of your homeworks for the next set, which will be the next module, which will be chapters 8, 9, and 10. 8's kind of an introductory chapter. Pay particular attention to the note outline because there are parts of 8 that you do not need to know. And don't waste your time studying them, even though I get it. You want the best understanding that you can have possible of a particular topic, but you can't understand everything to the nth degree just in this class. So for chapter 8, pay particular attention to the parts that are covered in the note outline and the video or videos. I don't remember how many there are for chapter 8. And then chapter 9 is probably the hardest of the set. Spend the most time on 9 because if you understand 9 pretty well before you start looking at photosynthesis, photosynthesis is going to be super easy because a lot of it repeats from chapter 9. So my advice would be chapter 8, don't overstudy the particulars that are not important. Focus on what's on the outline and what's in the lecture video for chapter 8. Spend your most time memorizing, organizing, making flashcards, practicing dynamic study modules, whatever you do for studying, spend the most time on chapter 9 and do it early so that chapter 10 is easier to understand. That's my advice. Okay, I'm going to close this then. Other questions, comments, worries, concerns? All right then, I'm going to end the recording and I do apologize. I didn't post Monday's recording, so I'm going to post that one and this one. It usually takes about 15-20 minutes for it to process, and then I post up to YouTube. So it'll be hopefully before lunchtime. I'll get the one from Monday and from today up there so you can take a look at them if you want. Monday's was pretty short as I remember. So today was obviously much longer. Much of this was covered in the Q&As that are up there for chapter 6 and chapter 7 from the fall, because these lectures I'm pretty sure were pretty extensive from summer. So I think I reused the summer lectures and then just did Q&A that was long, like today, about like hypertonic, hypotonic, and maybe some of the structures of the cell. I kind of forget. All righty then. Other questions? Other worries, concerns? Yes, no, maybe so. Not really. All right then, good luck on the quiz. Like I said, it's 45 seconds a question. The questions do not automatically move from question to question, but if you try to look up the answers to the questions, you're going to run out of time. So my advice is study as hard as you can before you sit down to take the quiz, and then do your best. Remember, if you take all of the quizzes, I believe there are 8 of them. If you take all the quizzes, your lowest score will get dropped at the end of the semester. And for some people, this first one's going to be the lowest score. For other people, it'll be somewhere in the middle, but it just kind of depends on your skill set, what you're coming with, and such.